neuroscience, cognitive neuroscience retreat, a fellow gave a great talk about magnetic sensing. And he decided to give a talk today on a completely different topic, which I think will be of interest to a lot of people here. It's near-infrared spectroscopy, which is another way of measuring brain activity, and it's basically the same blood oxygenation level dependent signal, but with a measure with a very different method. But it allows studies in different populations like children. It's more portable, and it was probably the technology that was used in the Minority Report. Although they use blue lights in the Minority Report, and actually it's supposed to use near-infrared light sources. Sal got his bachelor's in engineering here at Dartmouth, and then went on to Harvard, where he got his PhD, and then he returned back to Dartmouth, where he is now tenured in the Thayer School of Engineering, and he is working on this suite of state-of-the-art methods for neural measurement, which really could be the future of how we measure brain activity in a very fine-grained way. And so he's someone that we should keep track of for what he will be producing and innovating in the near future. You ready?
Thank you, Jim. It's a very kind introduction. Thank you so much. And thank you for having me here today to speak at your symposium. I'm going to take the afternoon to introduce you, for those of you who are unfamiliar, to this neuroimaging method called near-infrared spectroscopy. And also to talk about how this technique can be combined with other methods of neuroimaging like fMRI and with EEG to get additional information about the brain. So let me give you my outline. So I'll give you a little bit of motivations for multimodal neuroimaging and introduce you to NIRS systems and NIRS data analysis, hit some of the frequently asked questions about near-infrared spectroscopy, and then discuss the multimodal combination with EEG and fMRI systems, and give some examples of data analysis. And this is a lot to cover, so I'm going to move pretty quickly through this. And my understanding is that I've got 2.5 hours, right? Exactly, right. Okay, so I'm going to use every minute of that. Okay, so what's the big deal with multimodal neuroimaging? So if you think about measuring human brain function, there are several different types of signals that you might be interested in. You may be interested in neural activity resulting from action potentials from synchronous activity of neurons in the brain. You may be interested in something that is metabolic related, something to do with the energy demands of those cells in the brain when they're active, or hemodynamic activity, where you have a coupling between the neuron activity and the blood flow and blood volume response that gives rise to the fMRI bold signal that you exploit so frequently in this building. So there's a range of different techniques that one could use to measure these types of signals. Here's on our horizontal axis, we've got a temporal resolution ranging from sort of millisecond scale on logarithmically up to second scale into minutes and so forth. And then on a spatial resolution axis from single neurons up through columns, maps, lobes, and the whole brain. And each method of measurement lives somewhere in this space, and these are circles and ellipses and boxes that come out of Bin He's review from the IEEE Transactions on Biomedical Engineering a couple of years ago. So that one could debate about where exactly these belong on the plot, but there is this general idea that you have different combinations of spatial and temporal resolution available to different techniques. So something like fMRI tends to be more on the second scale, or a couple seconds per frame of acquisition, and then you're sort of in this zone of mapping, maybe down to columns. The techniques like EEG or MEG, the magnetoencephalography, have a nice fast temporal resolution, but the spatial resolution tends to be fairly low, unless you can assume that you have a known number of sources that you're just trying to find within the brain and locating like if you know there's one source and you can find where that is pretty accurately, but if you don't really know what you're looking for, the resolution tends to be very low. Near-infrared spectroscopy tends to be a little faster than fMRI and lower resolution. So its resolution is more comparable to the EEG and MEG, but your time resolution is faster than what you're familiar with fMRI. And there's some other differences that'll show up as well. But what a lot of people would love to do is to take these techniques and bring them together into a multimodal approach that gives you higher spatial and temporal resolution at the same time and get a better understanding of what's happening in the brain. So it sounds like a straightforward proposition. In the details, it's not so straightforward. I'm going to be speaking today a bit about EEG. And so just to get you thinking about it, here's a picture of a EEG cap with some electrodes on a mannequin here. And generally speaking, we're looking at signals arising from synchronous neural currents in the brain, very high temporal resolution. It's a non-invasive approach. All the techniques I was showing there are generally non-invasive approaches. And this is used clinically in things like epilepsy, sleep disorders, and so forth. Near-infrared spectroscopy is it's a medical monitoring technique. It's also just a functional neuroimaging technique. It uses laser light to measure concentration changes in your hemoglobin species, of oxyhemoglobin and deoxyhemoglobin in the brain. So the basic idea is that you're going to have some location on the scalp where you have fiber optics carrying near-infrared light and delivering that to the surface of the scalp. That light is going to propagate through the tissue, and we'll get into a little bit more details about that propagation in a moment. Some of that light that goes into the head is going to get detected by another optical detector. We call it optode. So it's going to gather up some of the light that's dwelt down inside of the head. And we're going to do some analysis then to figure out what happened to these concentrations of oxy and deoxyhemoglobin in the head, and hopefully resolve that down to changes that occurred in the brain. So let's tackle this by thinking about first the hardware systems. So you've got, in general, the goal is to measure oxygenated, deoxygenated hemoglobin concentration. And we're going to infer that from the detected light by using some different mathematical calculations. The one that's most commonly used is called the modified Beer-Lambert law, which is very much related to classical photospectrometry, where you put a solution in a cuvette and drop it into a machine, shine some light through it, and look at the absorption spectrum. When you take that out of the controlled geometry and you have scattering light, now you have a problem that is much less well constrained. And so the laws have been modified, so it's called the modified Beer-Lambert, and that's what we use. And I'm not going to go into the mathematical details of it, but that's the technique that's common. Fundamentally here, we need to understand that there is an absorption spectrum that we need to think about to understand how the light is absorbed differently. And so here I've got wavelength on a horizontal axis running from about 650 nanometers up to 1,000 nanometers. On the vertical axis, I've got absorption coefficient. So this is, this absorption coefficient is telling us how likely a photon is to get absorbed as it moves through this medium. And so a higher value here would mean that it's more likely to get absorbed. And we've got three lines here. Water is in blue. We've got deoxyhemoglobin in green, and water's in red. Excuse me. Water's the red line here. The oxyhemoglobin is in blue, and the deoxyhemoglobin is in green. So there's this window, we call it an optical window, between about 600 or so and 900 nanometers, where the absorption by water, that red line, is pretty low. And the absorption is dominated by the hemoglobin. And not only that, but the spectrum is different for the oxy and for the deoxyhemoglobin. So if you send in to the system at least 2 colors of light, and you assume that the changes in absorption over time are due to changes in the concentration of your oxy and deoxyhemoglobin, you can back calculate out the concentrations of those two species. So that's the idea of the spectroscopy. It is important to choose wavelengths that make this calculation work out well for you. I say it's a minimum of 2 wavelengths because you're trying to resolve this oxy and deoxyhemoglobin. Water, you can often assume, is a constant background. And so if you set everything to a 0 baseline at the start of your recording, time varying changes there could be attributed then to your hemoglobin species. There are some problems that come in. Just to mention one of them, we call it chromophore crosstalk, and that's where you can have, if your light propagation model has some inaccuracies, you can end up in your estimate, mapping one hemoglobin species to the other and vice versa. So you get a systematic error. We call that a crosstalk error, and that can happen particularly if you only have 2 wavelengths. So in my system, I use 4 different wavelengths. We use one that's at this isobestic point, right where the oxy and deoxy cross. So if you measure right at this wavelength of 808 nanometers, then changes in that signal level are related to the total amount of hemoglobin that is in the tissue and the changes in the total hemoglobin in the tissue. So that's a nice cross-check. So if you get an oxy and a deoxy, you add them together, you should get the same result as the measurement made at 808. And then I'll often throw in still a 4th color of light to help deal with noise issues from the instrument and give us a sense of confidence in our results. So when you put light into the tissue, there are only a limited number of occurrences that can happen. So if you have a photon going in, well, I'm not showing the surface interactions. You could have surface reflections and so forth, getting the light down into the tissue. Once you're down in there, then it might travel for a little bit and be absorbed. It could be transmitted, or it could be scattered. And that scattering could lead to later absorption, or could scatter and transmit, or could scatter and be re-emitted or remitted from the tissue. Cell phone interaction. Let's do this. Airplane mode. Okay. So there are different types of scattering, and I put this in for my colleague, Professor Brian Pogue, who has a background in physics, and he'll always ask when a student is starting to study near-infrared spectroscopy, what is the physical basis of the scattering, because there are different types of scattering that occur in tissue. And it depends on the relative size, generally, of the wavelength of the light and the particles and the things in the tissue that are with which the light is interacting. So there's this Rayleigh scattering when the molecules are in small structures or particles have a radius much less than the wavelength lambda of the light. Debye when they're larger but still smaller than the wavelength, and me scattering when the radii of the molecules and so forth are larger than the wavelength. And this me scattering is the one that dominates. So if you see Professor Pogue, you can tell him, you heard from Saul that near-infrared spectroscopy operates in this me scattering regime, and he'll do that. What? He'll be pleased. Okay, so a NIRS system at the cartoon level, and I should mention, so this slide and many of the slides that I'm showing here are compliments of my postdoc, Paolo Giacometti, who's there in the back, and smiling every time one of his slides comes up. And like many of you who do your research here, we rely heavily on our graduate students and our postdocs, and this is no exception. So thank you, Paolo, and I particularly like the hair on this guy. So the general idea is you've got these lasers, and you're shining light to someone's head, and you've got a source optode here. It's a giant suction cup. We make them a little smaller than that, in fact, but it's easier to see in the cartoon. These are optical fibers, and then there's a detector, goes over to a light detector. So those are the basic elements of the NIRS system. They also come in multiple different flavors. So you've got ones like we use in our lab most commonly are continuous wave, where you have a an intensity of light that is applied to the sample, and then you measure changes in the intensity of the light that comes out. And this light stays on continuously, and you measure continuously. So you can look at changes over time in that intensity level. There are other ways to do it. You could have a very, very short pulse of light and send that into the tissue and then detect back with a very fast sensor and measure what we call the temporal point spread function, or the TPSF of that light. And the broadening of the light pulse as it moves through the tissue carries all sorts of information about what's inside. And there's still another way where you have your lasers that are oscillating, usually sinusoidally, and you measure the light after it's gone through the tissue in terms of the amplitude and the phase. So you get a complex measurement, and you can again use that to infer information about the tissue. And the hardware that you use for each of these three is different. You have different laser configurations, different detection hardware, different processing of that data, and so forth. And you get a little bit different information out from each. So when you start thinking about near-infrared spectroscopy, when you're outside, it's just one thing. And you start to zoom in, and when you enter the field, there's actually a range of different techniques all within the near-infrared spectroscopy space. So the continuous wave, I mentioned this is the one we're using in our lab. And you basically measure a DC signal that gets modulated by properties of the tissue. It's the time resolved, just to zoom in. And the time scale of propagation here really is on the nanosecond to picosecond. You might use a picosecond laser pulse and measure it broadening out over the nanosecond scale. So these things are really fast. And the frequency domain here, one of the nice things about the frequency domain is that you can, that differentiates it from the continuous wave that we use. From the frequency domain, you can get the absolute properties of the tissue. So what I've been talking about so far is measuring changes in the concentration, always changes in the concentration of oxy and deoxyhemoglobin relative to some unknown baseline. If your interest really is in something like characterizing the tissue, let's say you want to do, if you're Brian Pogue, you're looking at maybe breast cancer and adding information to aid in the diagnosis and monitoring of cancer therapies. And you really want to characterize the tissue. Then you would use something like a frequency domain machine. The downside is that you need some fair amount of integration time to get those properties. So if you're doing brain function studies like I do, then you would choose the continuous wave instrument because you're going to get a better temporal sampling rate and capture those dynamics in the brain physiology more accurately. So if you want to do imaging, one of the things you're going to need to do is to have more than one laser and more than one detector, and you're going to start to get an array here. And so driving down cost and size also matters a lot there. So that's another reason we use the continuous wave approach. We often use a semiconductor diode laser. On the detector side, there are things called photomultiplier tubes, which are really fascinating if you like optical physics. Photodiodes are very common, a low-cost approach, but not particularly sensitive necessarily. These ones called avalanche photodiodes are quite remarkable semiconductor photon detectors, and they really have enabled some explosion of the availability of near-infrared spectroscopy for brain imaging studies because they're small, they're relatively inexpensive, and so you can have an array of these detectors. Other stuff you need fiber optics, you need lenses, you need headgear, so you need a bunch of stuff. We have a system that's made by a company called Tekken. They call it their CW6. It really was, this company and this instrument was born originally out of David Boas's lab at Mass General Hospital. So he works in the Martino Center for Biomedical Imaging. That's where I did my postdoctoral studies with Dr. Boas. And so he has been working on the science and the neuroscience and the physics of near-infrared spectroscopy for many years. And spun out of his lab and through collaborations and so forth is this company, Tekken, and they've been developing the instruments for quite some time now. So they're one of, I'd say, a dozen or so companies around the world that make near-infrared spectroscopy machines. This one has 32 detectors and it has 32 lasers, and we've got all the fibers and optodes to make this go. I think I've got a photo of it a little later on, but it sits on a cart. It's a box. Instead of having wires coming from it, it's got fiber optics coming from it, and so it's very reminiscent of an EEG system with a little larger box. And in terms of pricing, something like this would be in the several $100,000 range, which is actually not, well, so it's higher than EEG, but not necessarily. It depends on which one you buy. Okay, so that's a rapid overview of the NIRS systems. What about the NIRS data analysis? So I broke this down into 10 easy steps. So we're just going to characterize our signal levels, identify motion artifacts, convert intensities to optical density, characterize noise in each channel, remove low-frequency drift, perform our spectroscopic calculations of hemoglobin, remove additional physiological noise, construct a forward model, invert that to get tomographic images, and do our statistics. No problem. So the point of this is not, I'll take you through a couple of highlights here and there. The point is just that this is similar in complexity to fMRI analysis. There's a lot going on here. You need to understand something about the underlying physics to know what sort of data you're working with. You need to understand something about the underlying physiology in order to know how to sort out signal that is real from signal that is not, or signal that is from brain function from signal that is from other stuff like your scalp. And in order to get localization, you need to do some pretty sophisticated mathematics and computations to take measurements made at a surface and reconstruct it down inside of a volume based on these surface measurements. It's not unlike trying to find an oil field. A lot of the mathematics are actually similar, used in near-infrared spectroscopy as in geophysics and so forth, where you have surface measurements and you're trying to find out what's underneath. And furthermore, you have a hard problem because there's a whole lot of territory down in the brain, as you know. And even if you have the largest, most capable near-infrared spectroscopy system that is available, you're still going to have a small number of measurements relative to the size and complexity of the brain. So that's part of what keeps the resolution low, and it's part of what makes the math hard. Okay. So one of the ways that that we improve the localization of NIRS is by working from good anatomical models, and those, of course, are going to come from MRI. So this was one of my a recent graduate of the lab with a PhD, Catherine Perdue, who's now at Boston Children's Hospital doing near-infrared spectroscopy with babies, which is a great population to study with this machine because they can wear a small cap and it's can conform to their head, relatively lightweight. They don't need to be inside of the bore of a magnet. So one of her frustrations was that by and large, the world of neuroimaging focusing on fMRI has not paid enough attention to the tissue outside of the brain. Because I mean, when you're dealing with an MRI signal, an fMRI analysis, you're able to strip away everything else and analyze what you're interested in on the brain. Now with near-infrared spectroscopy, you don't have that luxury. You have to deal with the fact that the light had to traverse through the scalp and the skull, down to the CSF, to the brain, and then back again. And so your data analysis has to take into account all of the tissue of the head between your sensors and the region of interest. So you can't just strip it away. So we found like a lot of the available algorithms for segmentation were not particularly accurate when it came to things like skull thickness and scalp thickness and CSF layer, and so we did some improvements on that, and it gives us a segmentation that we can work from for our data analysis. So what is this idea of a forward model for NIRS? This is just helping us to understand the physics of how light that is shined on the scalp, say here up on the crown, propagates down into the head. And what's been shown here is actually a continuous wave illumination thresholded at different levels, so that you can see at probably different decades of loss of intensity, how far down into the head the light is propagating. And the real reason why this works at all is that, just to remind you, that the water absorption is lower, and the tissue becomes fairly highly scattering to these photons. So they bounce around and spread out. So to a reasonably large degree, you can treat this as a diffusion problem of light diffusing down into the head. And so for us, the forward model means building up all these simulations at this point. So we take a real head, and we simulate the propagation of that light, so that we can then make inferences about where the light went in a particular person's head when we make measurements. And the light doesn't just go down in, we also have to detect it. And so if you take and you put a detector nearby to a laser source, now you're going to detect light that is in the vicinity of that detector, but you're detecting light that has originated at the source. And so you take a mathematical product of those two zones of influence, and you get something that looks a bit like a banana. And so it is called the banana function. And it's conveniently, thank you, Paolo, for making it yellow, so that it is a banana function. So this is what the sensitivity map looks like for NIRS between a source and a detector. And so you'll notice a couple things. One, it depends on where you decide to sort of threshold your colors in the plotting, but you do get down to the brain. You do get into the cortex, but you also go through the scalp and the skull and the CSF, as I was talking about. So you've got this signal that is a mixture of these different of chromophore concentration changes in different tissues that get combined together. Now, if you cover the head with many sensors, many source detector pairs, and say, what are the limits of sensitivity of this method, you get a map like this. This was also Catherine's work. And so we wanted to know, what regions of the brain are within the range of study for NIRS and which ones are not. And we've shown graphically here an average for, this is for one particular subject where we took a high resolution MRI and processed it through and thresholded it at a signal to noise level of 1 or 0 in dB. And so what you're getting is maybe a 1 to 2 centimeters down into the brain. And we went through and tabulated this for anatomical and functional regions. So we've got data tables that'll tell you in a general adult population what percent of this structure is going to be visible to NIRS, and in what portion of the population do we think it's going to be visible. So there'll be some regions where it's quite good, and others, if you're trying to get down deep into the temporal lobes or something, forget it. If you're trying to do, primary sensory motor work or primary visual, then this could work well. What do the signals look like? This is a typical time course of a hemodynamic response function measured with NIRS, where you have a period of stimulation blocked in yellow here, and you have some decrease in the deoxyhemoglobin and an increase in the oxyhemoglobin, and this is the typical. Actually, maybe the next slide looks more typical, but this is a fairly commonly observed response of the brain to functional activation, where the influx of freshly oxygenated blood more than exceeds the metabolic demands, and you get this excess of oxyhemoglobin in the region that has been activated. So it's the opposite of what you would expect if you were just thinking about it in terms of if you had a steady flow of blood and metabolic demand started sucking on that. This is going the other way. This is an active overcompensation, which, for reasons I don't understand, is the way that we've evolved to operate. Here's another hemodynamic response function with our oxyhemoglobin going up, our deoxy going down, a little bit of time lag from the onset of stimulation, and some lag in the recovery after the stimulation stops. This measurement is taken on a small child, and so this shows you what the headgear looks like, that there's a pad of sensors and of sources and detectors on the head, probably in a foam pad that's just Velcro strapped on. And then here, a small tomographic image has been reconstructed locally, showing the response of the functional response that's been measured here. So this would be a regional measurement with near-infrared spectroscopy. So let me hit some of the common questions that come up about NIRS. So I've drawn these from a review article that I thought did a nice job of summarizing issues that come up. And this particular article looked at 100 or so, I think they started with 180 NIRS papers and picked about 110 to look at in more detail. And they were focusing on the primary motor cortex activation. Many of these studies use simple finger tapping tasks or hand squeezing. It's interesting, a lot of the NIRS work for many years was just the sort of same old paradigms, just getting the instrumentation and analysis and everything working well. You see now more and more, if you go to conferences focused on NIRS, or if you go to a conference like the human brain mapping, for example, you'll see more and more studies looking at cognitive function, looking at medical conditions, and so forth, using NIRS. So it is breaking out of these simple designs, but this review focused on simple. So typical time course of response, and here you start to see that there is different information. So the time course of response in the oxyhemoglobin, which is something to which you are not particularly sensitive with an fMRI bold. It's not. fMRI bold is more related to your deoxyhemoglobin that's shown here. But you get there's different physiology in the oxyhemoglobin. And there's some variations depending on the task and so forth in the shape of that response. So is this response localized and regionally specific? So in these M1 tasks, yes, it is pretty well localized to M1 and to the appropriate contralateral hemisphere. You do get a little bit of activation sometimes in the ipsilateral side with a sort of motor task, and that's not too surprising. Do you have specificity to the oxy and deoxyhemoglobin chromophores? And how does that relate to localization? So yes, it seems like this does generally work out, and it's labs that have been doing this for more time will have a will do a better job of sorting out the chromophores. And it's interesting that you actually get a little bit different, sometimes you get a little bit different localization in your oxy and your deoxyhemoglobin. It may have something to do with the vascular structure down in the tissue there. It also may be just that your oxyhemoglobin is more sensitive to systemic effects, things like your cardiac pulsations and the blood pressure fluctuations associated with variations in your heart rate and so forth. A lot of these regulatory processes show up in the arterial side and then get damped out through the capillaries. And by the time you get to the deoxy side, you have less of this systemic physiology. So I always maintain some skepticism because it could be just that you have more physiological artifacts from the oxyhemoglobin. Then again, you also sometimes get a much larger signal change from the oxyhemoglobin. So if you've done a good job of cleaning up that signal, it might actually be a good localizer. Are these signals reliable and repeatable? Not too many studies have looked at this formally. There was one mentioned in this review that found a correlation of 0.84 for doing a spatial temporal test, retest repeatability. This is an area where I think the field could actually benefit from additional background rigorous testing and validation. But that's a hard grant to get funded, is the reality of it. But yeah, there is a fair amount of, you will find some variability as you go from lab to lab and looking at results. What is the correlation between these fNIRS signals and other neuroimaging data? So deoxyhemoglobin, abbreviated here HBr, is the one we would expect to correlate with fMRI bold. Given what's known about the bold signal contrast and things like the balloon modeling or Windkessel modeling of the hemodynamic response function, this is what you would expect to see. And there have been some studies. I'm part of this one by Huppert in 2006. Others have found good correlation with fMRI bold, but not universally. And reasons for that could range from variations in the instrumentation analysis, experimental design. There's not a lot of consistency in the field, so it's hard to know exactly what to think. When I was involved, we found good correlation with bold. Can this be modulated? And this is an interesting one. I think if you are doing fMRI bold analysis and you are assuming that your HRF is a constant and you're putting that into your general linear model and then say go. What we find in the NIRS measurements is that the hemodynamic response can be modulated by the task and by in this case with simple hand tasks, the frequency, the intensity, the complexity of the task changes not just the total response, but if you were to try to deconvolve that into its fundamental response function, there is some difference in that response function depending on the nature of the task. So if you have some differences in the physiology and you blindly plug ahead with your analysis without taking that into account, that could result in apparent statistical differences that are related to the underlying physiology, not necessarily what you might have had in mind. So this is the sort of area where NIRS could be complementary to help understand the underlying physiology. Is optode positioning reliable and accurate? So this is one of the areas of variation among different labs and different NIRS systems, is that you might be using your optical source and detectors in different places with different densities of coverage, with different amounts of light, with different wavelengths of light. And even just the positioning can lead to significant variations. So as you'll see in some of the upcoming slides, we've spent a fair amount of time in our group trying to get accurate positioning and repeatable positioning of these optodes. Okay, so let me transition into the multimodal perspective here. So this slide you've seen before, and now I'm hoping if you look at this again, you're thinking, okay, this goal of getting high spatial temporal resolution sounds good, but as we started to look at the details of this NIRS technique, it's complicated. It's not just a simple black box, plug it in and go. And so what are some of the details you need to consider? One is how you're going to put everything on the head. And I'm going to take you through some of this in the context of a study that we've got underway. I think I saw Heather in the audience here. So Heather Wishart and I are working together on a project sponsored by the DoD to study neurovascular coupling in patients with MS. And the idea here relates to my comments earlier about, well, what if you've got uncertainties in the hemodynamic response function? People have been studying MS, functional activation in MS for many years using fMRI techniques and have typically assumed that the HRF is a constant. But the disease is an evolving process, and it may alter that relationship between neural activity in the hemodynamic response over time. So then if you see changes in your map, is that due to reorganization of brain activity, or is it change in the physiology, but the neurons are still doing the same thing, or some combination of both? That's not something you can resolve with a single modality technique, but it is something we believe we can resolve by combining EEG, NIRS, and MRI and fMRI into a single study. This probe is covered with wires and optodes. This is the top of my head. This is an earlier version. I've got a couple other shots of it here in photographs. So we're in the process of redesigning this right now, specifically for the MS study. But this, here I am. Here's the NIRS machine, a box with lasers and detectors, a computer, and a monitor. It's pretty much all you need. And the EEG system's just in the background. It's a small amplifier. This head probe was designed by Paulo as part of his PhD work, which he did before staying on as a postdoc. This is some of the prototypes that Paulo built in sort of iterating through, trying to figure out how we're going to get accurate positioning and repeatable positioning in good conformity to differences in head geometry and so forth. Right now we're at the stage where we are trying to optimize the layout for the MS study, where we plan to use an N-back task for working memory, as well as probably some simple hand tasks or stimulation of the median nerve, so probably some primary sensory and other tasks that would involve some frontal involvement and so forth. So we're right now just trying to map what we expect to see from the fMRI and trying to position our optodes over those regions so that we can design a good study. But this is work in progress, so we haven't finalized anything yet. But here's a top view of like one of our candidate layouts. You've got EEG electrode positions in yellow. We've got NIRS source laser source locations in red and detector positions in blue, and we're trying to move these chess pieces around to achieve our best aim. There's also EEG system here at Dartmouth. Dr. Barbara Jobst over in neurology has a brain amp fMRI-compatible EEG. You may also have one here in Moore Hall. I'm not sure. Essentially, it's fancier amplifiers and filters and such with the ability to precisely tag the MR sequence into the EEG data file, so that you can try to remove artifacts after the recording is done. So the cleanup is all done in signal processing. So we're actually integrating Dr. Jobst's EEG system with our CAP so that we can have the option of making these measurements during fMRI scans as well. The optical measurements, just so you know, I mean, you've got fiber optics. Those are not going to get interfered with by the magnetic field. So there's really not a big issue besides just fitting stuff on the head into the bore. There's no physical interaction between the MR system and the NIRS system the way that you do with the EEG. This was our evaluating repeatability, precision, and accuracy of our head probe, and it works great. This is some software that Paolo developed, recently published in Neuroscience Journal Neuroscience Methods, where you can take an MRI scan, develop a mesh from that, pick your fiducials on the head, and it'll give you all the detailed coordinates for your data analysis. So not only do you need good positioning on the physical head, you need good corresponding positioning in your analysis. And so we've been developing those tools. This was an early experiment that we ran with the EEG NIRS head probe, where we were stimulating the median nerve. So this is something where if you are just at rest, you have a rhythm in your sensory motor cortex called the mu rhythm or the sensory motor rhythm. It's sort of in the 8 to 13 hertz range. And if you begin moving or you start to stimulate those nerves, the rhythm is silenced or it becomes desynchronized in some way. And then it comes back again when the stimulation stops. So it's a nice task for us doing EEG NIRS because the stimulus is very precisely timed in the EEG because we can electrically control the stimulation of the nerve. And you also get a hemodynamic response to the stimulation. So this was just showing that we have some localized, in this case, bilateral activation in the EEG, and the NIRS signals. We've got some hemodynamic response, and we've got this is a time frequency power map. So we've got this higher power mu band, and then on all of our EEG channels it goes quiet and rebounds. So things are just generally working. Okay, so some of the challenges in multimodal data analysis. So one of them is that the sensitivity that you have to the brain from NIRS and from, say, EEG is not the same. Even though you can put the sensors very close to one another, the physics is completely different. In one case, you're sending in light and scattering, you're detecting that light again. In another case, you're measuring the potentials, electrical potentials on the scalp that have originated from neural activity down in the brain, and that the spreading of that potential field is different from the light propagation physics. They don't decay at the same rate, and so you don't get the same sensitivity. So this is one of the challenges, and so this is Paolo's work again. We've been looking at the correlation between the sensitivity functions you get different locations on the scalp from EEG and from NIRS to then inform our later analysis. Because if we were to assume they come from the same underlying tissue, we would be very wrong. So we're going to have to take into account that we have, in the range of 0.6 or so correlation over our sensory motor area for the NIRS source detector pairs grouped in certain ways versus electrodes. If we map that down onto the cortical surface, you can see that there are hot spots on the gyri, and there are cold spots down in the sulci where the correlation can be reasonably high or fairly poor. And so one of the things that we've realized is probably important is to do this as a region of interest analysis. So what we did is we did an optimization study where we said, okay, suppose you had different blobs for a region of interest, and you scaled those blobs to be different sizes. At what point do you get the best correlation between these two sensitivities? And it sort of gives us the sort of the least common denominator for joint analysis. And it smears out a lot of those variations we sacrifice some of the spatial resolution, but we improve our ability to make joint inferences. This is making that point a different way. We can, in simulation, say, suppose we had a very small functional activation on the cortical surface. So we make a little red dot where we're going to say, okay, we've got activation here. And then we can simulate what that measurement would look like in a world of perfect knowledge for NIRS and for EEG. So we get a measurement set from each. And then we try to reconstruct that source using inverse methods from NIRS and from EEG. And we see, well, how do those reconstructions compare? And this is what we get. So we call this a point spread function. We started with a point. We push it through all the physics of the instruments and the measurements and the analysis, and it spreads out. So this point spread function for NIRS looks something like this. And the point spread function for EEG, it's a different shape. And so that's fundamentally at the level of the physics and the analysis. We can't do better than this in terms of getting common sensitivity. Now they correlate, but it's not perfect. All right. So there's another layer to this. That's just sensitivity on physical space, but there's another layer, which is physiology. These signals that give rise to the EEG and the NIRS are not the same thing. They're really different. And the thing that ties it together for us neuroimagers is this idea of neurovascular coupling. So this is where this is the relationship between neural function and the vascular hemodynamics. And so generally speaking, as we've seen in the hemodynamic response measurements I showed, you've got this increase in blood flow of oxygenation that follows the stimulation. And what gives rise to it is complicated anatomy and physiology. And I'm not going to try to go into details here, but this is a nice paper by Ayatakola from 2004 looking at neurovascular regulation and neurovascular coupling in Alzheimer's disease. And but basically the key players are we've got neurons, we've got glia and vascular cells that are interconnected and signaling to one another so that you get a relationship between that neural activity and the hemodynamics. So it's mediated through a complicated set of anatomy and physiology. If you zoom in even more, and you've got you start to look at the different chemical signaling that is going on between between your neurons and your glia and the way that those are going to connect through to your vascular cells and affect the dilation of your arterioles and affecting ultimately the blood flow and blood oxygenation in the tissue. From a simplified cartoon level, we've got action potentials. We've got extracellular ionic currents involved, vasoactive ions, so there's some sort of other ions that are signaling the dilation of the vessels, and ultimately leading to your vasodilation, which all takes time and isn't necessarily going to be the same in every person, in every part of the brain, and in young and old, and so there's a lot going on. And the types of factors that are implicated in the signaling and neurovascular coupling are many. So there are different ions, metabolic factors. There are neurotransmitters that are active here, other things that are all, the nitric oxide tends to play a key role here. So there's a big complicated picture that relates these two signals physiologically. One can try, and I have tried, to model all of this mathematically. You can pull out your mathematical physiology books, and I did a study in 2009 looking at the blood flow and gas exchange and dynamic cerebral autoregulation component. And I found another study by Aubert, published in PNAS in 2007, that does a similar thing for the neurons in the astrocytes, capturing the dynamics of the metabolism And I tried to plug these together, and I could get the code to all work. At the end of the day, I've got like a mathematical model with on the order of 150 or so parameters to keep track of, and I've got no idea how to get all those right. So it was interesting, and I learned a lot, but I don't know that it's particularly useful. Another way to do it is much more of the engineering black box approach, where you say, okay, well, we believe that primarily the neuronal activity drives the hemodynamic activity. And so let's take and find some features of the EEG. In this case, we're using the power envelope, which is time-varying power envelope. Convolve it with some kind of transfer function that we're going to fit so that it predicts as well as you can the NIRS signals that are observed. So we've got an observation here in EEG, an observation on NIRS, and we're trying to find the system model that links them up. So you pick a class of functions you're going to use to fit. In this case, we're using a gamma transfer function with a certain amount of flexibility. And it's more black box, not completely black box. I call this a gray box, maybe. Because you get some insight. It ends up working out for a certain set of transfer function parameters. There's probably a relationship between those parameters and the underlying physiology, which might be different in young or old or in different disease states. We might not be able to pin it down all the way to the level of what ions were involved, what neurotransmitters were involved, so we don't have that data. But we might be able to get a statistical difference that could be useful or provide some insight. So that's some of the challenges. Let me summarize. So NIRS, so it measures, generally speaking, changes in the concentration of oxy and deoxyhemoglobin with good time resolution. The spatial resolution is more like EEG. So this is so it's good for studying hemodynamic physiology associated with brain function. And the key for me is the physiology piece. If you want to know what's going on with the hemodynamic physiology, this is a great complementary technique to use. NIRS can be integrated with EEG and fMRI for multimodal neuroimaging experiments. Its integration with EEG outside of the fMRI is relatively straightforward. Its integration with fMRI by itself is, again, relatively straightforward. Putting all three together is definitely a challenge. But it does open up the door to studying things like this neurovascular coupling relationship, which takes the physiology a layer deeper. But you've got to deal with a lot of complexity. And so at the end of the day, the promise of using your NIRS and your EEG and your fMRI to get higher spatial temporal resolution, I think is a false promise. I think at the end of the day, what you're going to get from this is more insight into the brain physiology, not better resolution, because you've got all these other issues I'm bringing up about overlapping, mismatched sensitivity and complex physiology and so forth. Developing the analysis methods for studying neurovascular coupling with NIRS, EEG, fMRI, it is an active area of research. And it's active both on the side of mathematical computational methods and on the side of understanding the underlying physiology. We don't know enough about the brain and brain physiology to just write down the equations and fit them. We don't know which are the primary drivers of neurovascular coupling from that long list. We know that many are involved, but to what extent and exactly how. Those details are not really known. And that makes the analysis all the more difficult. And so you've got this double unknown, and so it's an active area of research to try to bring that together. And let me acknowledge some of the other contributors to this work. In the Multimodal Neuroimaging Lab, Paolo is here, Jenny, Gina, Priyanka, Brad, Aaron, and my administrative assistant, Ellen, Werta. We all have contributed in various ways to work that you've seen here. Specifically on the NIRS EEG project, Heather is here and we've got other collaborators from the Geisel School also working with us on this project. Funding specifically for the EEG NIRS has come from the National Institutes of Health, the Department of Education, Department of Defense, Newcomb Institute, Dartmouth IQBS. So it's been supported by different groups in different ways. And let me also just mention that we are dealing sometimes with issues that arise from power outages. It's no fun. Went out like 5 times in a week. Burnt out component on our main board. And these are some of the references. I tried to give most of the references along the talk. These are the ones that didn't get referred to directly. So thank you very much for listening. That could be achieved with NIRS. Right. Yeah. So there is evidence that you can get resolution usually I say 1 centimeter scale at best, but Joe Culver from University of Washington, St. Louis, has demonstrated that you can do things like you can get with very, very high density coverage over the occipital lobe, you can get the same sort of pinwheel analysis of a visual activation with potentially even higher resolution than you would get from a typical fMRI. But it's like the entire instrument's been designed around that experiment. So it is possible, but it is usually for the typical user, it's sort of centimeter or so resolution.
And then on your temporal versus spatial resolution graph, yours has got higher temporal resolution.
That's right.
Even though it's the bold signal, which has inherent limitations in temporal scale. But that's because of more frequent sampling. It's really.
That's right. So there's really no limitation for how fast you could sample with the NIRS. You just want to go faster than anything that you're measuring in the physiology. So the fastest thing in your hemodynamics on the oxy side is usually your cardiac pulsations. So you can get, with a good NIRS system, you're going to get probably 6 to 10 harmonics of the cardiac pulse in your data, because you're going to end up sampling at, you might be sampling at 25 hertz, or maybe you're going to go at 100 or more. So is that faster than you need for the deoxy response? Yes. Is it important if you want to capture the onset time and peak time and decay time constants? Yes. Yeah.
So you mentioned some of the studies that we're looking at infants or young children, looking at sort of brain activity and development, but it's hard to stick them in the MRI, for example. Are there differences in signal quality? Is it just sort of different parameters that you input there, or is it?
Yeah. So you get better signal quality from infants and from children. You can actually transmit light right across the head of a baby, which you could never do on an adult. So that gives you higher resolution spatially. It gives you just a lot of advantages. So it is a good technique to use on children. You do, though, have motion artifact issues, which you've got to deal with. And so, children aren't quite so good at staying still. So there are some trade-offs. Other questions?
Are there any safety issues at all with this?
They're minor. You want to keep the lasers off until the probe's on the head, and then turn them on and turn them off before you take it off the head, because someone might say put a laser light into their eye. It's a similar intensity to a laser pointer, so you could cause some eye damage if you were really careless, but some just general good safety practices, and you're not going to warm the tissue. You're not going to cause any sort of it's a non-ionizing wavelength, so there's very minor issues. Other questions? Okay, thank you all very much.